Thank you for attending today's virtual seminar, Modeling and Measuring Carbon Assimilation by Plants, presented by Dr. Colin S. Campbell, Vice President of Research and Development at Decagon Devices. Dr. Campbell.
Thank you, Jordan. I'd like to welcome everybody out here to the seminar on modeling and measuring carbon assimilation by plants. This is actually work that was put together not only by myself, but several of my colleagues here at Decagon. My father, Colin Campbell, Steve Garrity, and Doug Kobos. And I've gone through and put together several slides out of some of their presentations so I can try to combine information today that will help you learn more about measuring carbon assimilation. And I hope by the end of this, you'll understand especially the newer techniques that we've been working on here that will help you in this process of measurement. So let's first just briefly go over an outline of what we're going to talk about today. I'm just going to give a brief introduction to resource capture. Then we're going to go on and talk about measuring carbon dioxide directly. Now, admittedly, I'm going to go back and grab some of my old data out of a project I did at Texas A&M University and talk specifically about how we measured carbon dioxide uptake or biomass accumulation there, because I think it's particularly relevant to the topic as we move on and talk about some ways to monitor that, especially by radiative techniques. And that's really going to be our focus today, as we're going to talk about simple resource capture models and how we might go and use current techniques and just techniques that are just coming out to measure in the field biomass accumulation and radiation use efficiency. And we're going to just drop down through that list, talking about total solar radiation measurements, fractional interception measurements, and then radiation use efficiency. And then right at the end, we're going to say, well, what can we do with this, and what does the future hold, and where do we need future research to spend time looking at areas. Dr. John Monteith, back in 1994 paper, described a resource as a form of energy or matter that plants need in order to grow or reproduce. He defined capture as the process by which any organ in that plant above or below ground removes a resource from its environment to maintain its metabolism. And these are pretty important definitions as we go and talk about resource capture. Now, the plant resources that we're going to focus in on today are carbon dioxide and light, or solar radiation. In other seminars, we've talked about water as a particularly important resource. That's a big passion of mine, but today we're going to leave that out of this particular seminar, and we're going to talk about that on another day. Now, nutrients we're also going to ignore at this point and maybe come up in another virtual seminar at some point. Why do we care? Why do we care about modeling resource capture? Well, really, it's all about the biomass accumulation, the carbon dioxide uptake. Carbon dioxide, being a greenhouse gas, is particularly of interest right now to the science community. And many, many people are out there trying to look at different ecosystems and their source-sink relationship for CO2. I did that in my research, so we'll talk about it in a moment. Many, many people are doing that today, and so it's a particularly relevant topic. Now, other important things as they relate to biomass uptake are things like crop yield. We know the world's population is growing. We need to find better ways to feed that population. And finally, biofuel production, also very, very important in terms of our energy use. We can also talk about radiation use efficiency. Radiation use efficiency is critical as we think about how to increase yield for particular cultivars as we try to work in crop breeding programs to develop better radiation use efficiency in plants. And also, we'll mention a rather interesting study that talked about stress conditions and how stress conditions affect the predicted yield versus the actual measured yield that we get in the field. So let's spend a little time talking about directly measuring resource capture, CO2 accumulation in the field. This is a pretty well-known study that was done by Dr. John Monteith, again, back in 1977. He showed a very good correlation between total biomass accumulation, total dry matter accumulation, and intercepted solar radiation as it accumulated across a season. And he noted that these were linear related to each other, and they really depended on the plant, the crop, the ecosystem, that relationship. You can see that apples, barley, potatoes, and sugar beets there in the graph all show that linear relationship, but they're all a little bit different from each other. Now, it's always important to note in a graph like that where we're accumulating two variables, one on each axis, that there would be a correlation between any even randomly associated variables or random variables that we put together. But in this case, we know that there is a clear mechanistic relationship between this intercepted solar radiation and the total dry matter. So how might we start measuring the biomass accumulation in the field? I wanted to start with this slide because really if we wanted to do an extremely rigorous job of this, there are a few things that we can do, and I've got some examples here. The top left picture is my student worker many years ago measuring rice in an irrigated, flooded rice field in South Texas. And this was a commercial rice field where we were measuring eddy flux, and we'll talk about that in a moment. But to try to understand the correlation between our CO2 flux measurements and the actual biomass accumulation, Cody and I would take a 1 meter square PVC, and we would go and throw it as far as we could into the fetch area, meaning the area where the CO2 was being taken up, that was being measured by our flux system. We'd throw it into that footprint, or what we call the fetch, and we'd go and sample all the biomass in that 1 meter square area, the above ground biomass, and then take root samples. And then we'd go and analyze all that biomass to see how much there was in 1 meter square. And in that way, we got very, very good measurements of the biomass accumulation. Now, I can tell you that neither Cody nor I enjoyed that process very much. It was a very accurate way of doing it, but it also was very hot and very, very time consuming. So other people have thought up other ways to do this. One other note on that, if we go out and sample this biomass, you have to also understand that we're not going to be able to measure those plants again because we've harvested them. And so we're altering the system essentially that we're trying to measure. So people have said, well, how can we go out and measure these systems without altering them? One of the ways is a closed chamber, like I'm showing in the upper right-hand corner. That's an experiment in Australia where they've actually put out closed chambers and measured all the CO2 going in and going out, and so they can actually quantify the biomass accumulation, for example, in trees inside those very large enclosed systems. And the larger picture on the bottom is a eddy flux system. This was actually the one we used in Texas that measured the upward and downward eddies as they move past the sonic anemometer, and measured them with a CO2 analyzer. And this way we were able to get the CO2 flux into our rice field. Now, all of these are very, very good ways of being able to get at the biomass accumulation. But the problem is that, as we note there, they're altering the system. They're very time-consuming and often extremely complex, and the systems are always expensive. Now, what can we learn, though, from the data that we get out from these systems? Well, here's that picture blown up so we can look at it a little better. The 3D sonic anemometer I have labeled in that picture that you're looking at. That 3D sonic anemometer is just measuring the upward and downward eddies that flow past, and we split the up and down eddies into different containers so that we'd be able to measure the concentration of CO2 using conditional sampling. With our data acquisition system, we were able to measure and store the CO2 flux over time. And then we were able to create quite a large data set from 2 years in this irrigated rice. Now, we're not going to spend too much time looking at these data, but what we want to observe is this source-sink relationship that we have over the season in this rice field. So initially, we have a negative carbon dioxide exchange rate. Now, more recently, we call this the net ecosystem exchange or some other term. At that time, this was the common terminology, but it's in moles per meter squared per day. That's moles of carbon. And what you can see there is over time, so DOY stands for day of year, first through the primary crop and then through the ratoon crop or the second crop. Rice is a tillering plant, and so it would grow a second crop. It would continue to tiller after the first harvest, and you'd actually get a second harvest of rice later on in the season. Initially, it would start as a net sink. One of the reasons was that during some of that time, the rice field wasn't flooded, and so we had carbon dioxide emissions from the soil itself. Then we had flooding, and the rice crop growth was significant enough to be a net sink of carbon dioxide for quite a long time. I've labeled a couple of points in there: the panicle differentiation, which means the beginning of the fruiting structure on the rice, heading when the rice seeds began to fill, and then noted where that field was drained. At that point, the rice was actually senescing. We had considerable leaf die-off. So with this CO2 system, we were actually able to monitor the change in carbon dioxide uptake over a season. Now, that's interesting, but how does that apply to resource capture? Well, we're seeing resource capture of this rice crop plotted with daily values across that season, so that's quite interesting. But how do we know that we're in particular measuring the actual biomass uptake of this rice crop? So here I plotted 2 years of the study. So on the left-hand side, you see 1998, right-hand side 1999. So these data are pretty old at this point. But you see the relationship between the cumulative carbon dioxide exchange rate multiplied by 0.77 compared to the field biomass. You say, well, why did you multiply by 0.77? We sampled the rice across the season, and were able to analyze the rice after drying it and crushing it to get total carbon in the plant, and therefore we're able to make a conversion between carbon dioxide uptake and total carbon in the plant. And as these graphs show, we had a very, very good correlation between the CO2 flux that we were measuring and the biomass accumulation, which gives us a lot of confidence that we can then turn to the data and answer some of the interesting questions about resource capture. One thing that we noted was something very interesting that we're going to get back to later on in the presentation. This is a graph showing the carbon dioxide exchange rate in grams per meter squared, and then we divided that by our leaf area index on a daily basis, grams per meter squared per day per unit leaf area. And I've also plotted there the leaf area index, which is the total leaf one-sided surface area divided by a unit ground area. And so we see on this graph, the green line is just a model going through the black dots. Those are the measured leaf area index, so our maximum was right around 5. And then also graphed there in the red dots are this CER divided by the LAI. What you notice on that line is that as the leaf area index increases, we actually have a decreasing CER to LAI ratio, which means for the leaf area, we were doing less biomass accumulation as the season went on. Essentially, we were covering leaves with other leaves, and they were not as productive later in the season. Now, we turn from that understanding of what's kind of going on with the plant to look at a term we're going to get back to again later in the presentation, which is this RUE, radiation use efficiency. I also call this light use efficiency sometimes. And so we can somewhat use those interchangeably, but we always have to say what our units are that we're using. So in this case, we're talking about photosynthetically active radiation in micromoles per meter squared per second. Now, there is a correlation. We even noted this before. It was noted by that Monteith graph, that linear relationship. There is a linear relationship between the carbon dioxide exchange rate, or the biomass accumulation, and the intercepted PAR. That's not only on an accumulated basis, but as these graphs show, that there is a relationship even on a half-hourly average basis. So what I'm showing in these 6 plots is on the bottom, the photosynthetically active radiation, actually photosynthetic photon flux density, if we want to be absolutely correct in what we're saying. That is in micromoles per meter squared per second. And on the y-axis, we have carbon dioxide exchange rate. And I split these out for different leaf area indices. So we went on the top graph, the top left graph is less than 1 LAI. Then we go from 1 to 2, and you can see 2 to 4, 4 to 5, greater than 5. And then I put one in for post-anthesis because I think that was really important, is that rice head began to fill out. It changed the dynamics of the plant. What I did was then compare the slope of these lines, and there are 2 things that I want you to notice. First of all, the general linear relationship is not statistically different between the intercepted solar radiation and the carbon uptake between LAIs from basically 1 to greater than 5. Before LAI reached 1, that slope was significantly lower, and the post-anthesis, where we were seeing a lot of leaf senescence, that was also significantly lower than the other slopes. But generally, we can say that the radiation use efficiency was fairly consistent across the season. This was for that primary crop. Now, the other thing that we can see here is a rather interesting situation where we have a linear relationship between those 2 up to a point. And on some of those graphs, you can see a flat line at the very top. This is the light saturation in the rice, where basically we were increasing the intercepted light, but we weren't, in fact, increasing the carbon assimilated at that point. And the reason was that the leaves just couldn't do anything else, or couldn't take any more in. And we're in fact going to talk about that later as we get into the photochemical reflectance index, the PRI, and talk about how we can actually measure that with the PRI, with the spectral reflectance index. So let's take the next step and start talking about a simple biomass accumulation model. This is important in our discussion because up to this point, we've described techniques that were accurate, or very, very useful looking at biomass accumulation, but things that were too expensive to put in a lot of locations across the field. And if we're trying to understand resource capture on a large scale, it's simply impossible to try to set enough, let's say, of these carbon flux towers up to be able to do this everywhere. So what are some of our options, maybe less expensive options to do this? Well, let's get into this model that was proposed by John Monteith and talk about how we might measure it. So first, simple light capture assimilation model says that biomass accumulation is equal to the radiation use efficiency times the fraction of interception times the total solar radiation. And as we go down that list, the radiation use efficiency is really determined biochemically. It's a biochemical factor. The fraction of interception is determined by the plant canopy, distribution, the amount of leaves in the canopy, etc. And finally, the total radiation is just an environmental parameter that we can measure. Now, it's useful to note here, although we're not going to spend a lot of time looking into that the yield is just a simple multiplication between the harvest index, which is crop specific, and the biomass accumulation. So how do we measure these? The simplest one we can start with, which is total solar radiation. I'm showing a couple of pyranometers there. We can measure with any of these pyranometers, and there's lots of others available to choose from. We typically use an apogee pyranometer that is relatively inexpensive and also accurate. We can also use a quantum sensor. For example, those data I collected and showed a little earlier on our radiation use efficiency in the rice crop were all done using a quantum sensor because we were particularly interested in photosynthetic photon flux density, just in the visible wave band. Speaking of the visible wave band, let's talk about our next component, the fraction of interception. When light comes into a canopy, three things can happen to it. It can either be absorbed by the leaves, it can be transmitted through that canopy down to the ground surface, and it can be reflected, and that's showing right there. And one thing we need to note, and is going to be of particular interest as we talk about these radiative methods for measuring different plant properties, is that light is absorbed, transmitted, and reflected differently depending on the wavelength of that light. So how does that apply to measuring fractional interception? Well, if we had the canopy on the left, a very tall maize canopy, the fraction of interception is basically 1. I don't know that we'd need to spend a lot of time trying to measure the fractional interception in that canopy. And that was very similar to the canopy. Once I had rice completely mature, there was almost no light that made it down onto the water surface, the flooded water surface inside that rice canopy. But for most canopies, they're more similar to the picture on the left, where we have some leaf area with some soil that is also visible through that canopy. And in this case, we would need to measure fraction of interception. So how is this done? Well, the first method we'll talk about is measuring interception with ceptometers. And a ceptometer measured intercepted radiation, and that's where the name came from, as a ceptometer, is out of that the septed part of the interception. Now, a ceptometer actually isn't measuring interception, it's measuring the 1 minus the interception. So the fractional interception is equal to the 1 minus the transmission of light through the canopy. How do we measure intercepted light with one of these devices? Well, we're80 here the Decagon LP80 AccuPar ceptometer, and it's relatively straightforward. It's got 80 light sensors along that rod there, and these each are sensing the amount of light hitting that exact point. They're spaced at 1 centimeter apart, so that rod is 80 centimeters long. So we take a single light measurement above the canopy. This gives us how much light is actually irradiating the canopy. Then we go beneath the canopy and measure at several locations and get the transmitted light through that canopy. Our fraction of interception then is just 1 minus the fractional transmission through the canopy, and we can even convert this to a daily value depending on the needs, and that theory is all in the manual, in the LP80 manual. We also are interested in the leaf area index, the single-sided leaf area per unit ground area. The LP80 measures the leaf area index, but as we've noted earlier, that the biomass capture, the resource capture in terms of biomass, CO2, is much better predicted by the fractional interception. And above a LAI of about 5 meters squared per meter squared, additional leaf area doesn't capture any more light. And that we realized as we were looking at those radiation use efficiency graphs from the rice. And in fact, as it increased to a certain point, we had that linear relationship, but after that point, we had saturation, and really we had additional leaf area that just wasn't doing any good because we took up as much light as we could. So canopy, getting back to these canopy interactions, I mentioned already the three fates of radiation in the canopy, and that that light is taken up at different wavelengths differently. And in plants, we know this to be true. The visible spectrum is absorbed heavily by plant leaves, but the near-infrared spectrum is reflected highly by plants. And we're going to talk about that as it relates to things that may help us to measure fractional interception better. And so the take-home idea is that the interaction with wavelength or the light is wavelength dependent. And now I want to show you how this actually looks on a graph. This is a figure from Arroyo and Villegas, recently put out. I love this figure, and I asked him if I could use it in a presentation because it really illustrates quite well what happens to light as it comes on just some kind of crop canopy. What's happening here is there are several letters in that graph. H, which stands for heading, A is anthesis, M is milk grain stage, and PM is post-maturity. We also have one curve labeled soil there. And on the x-axis, we have wavelength. So we have the visible wavelength from about 400 to 700 nanometers, and above 700 we have the near infrared. And so what I want you to look at here is the changes that occur in the reflected radiation, which is on the y-axis, as these plants mature. So initially we have heading when the plant has the highest amount of leaf area and it's the most active. We have a very large difference between between the absorbed radiation in the visible range and the reflected radiation in the near infrared. And notice how that changes as that plant matures and goes even into post-maturity stage. And also note here the large difference between the soil spectrum and any of the plant spectrum. Now, if we were to measure the amount of reflected radiation in the near infrared range and compare it to the amount of radiation in the red range, you can see that there's quite a large difference when we talk about the heading point of the crop, and a relatively small difference when we talk about soil, or even talk about the post-maturity leaves where we've had a considerable amount of senescence. And not surprisingly, people have used this in a very common index, which we call the Normalized Difference Vegetation Index, or NDVI. And it's the difference between the reflected radiation in the near infrared and the red divided by the sum of those two reflectances. That gives us the NDVI. And there on the right-hand side of this slide, you can see some of the typical NDVI values. For example, bare soil, which is 0.2, a full canopy, which is around 0.9, a very actively growing canopy with few senesced leaves, and water, which is around -1. So if we were able to measure and compare the reflected radiation from the red and near infrared, we'd be able to have a pretty good index of the amount of leaf area. And in fact, that's really what we're going to talk about right now. Notice that to do this, we need to have our reflectance value, which is the ratio of the reflected radiation from the surface divided by the incoming radiation. And that's why I've got those two pictures up there. The one pointing up to get the incoming radiation, that's a hemispherical view, and the radiation or that's downwelling coming into the canopy. And then on the right-hand side, we have a field stop that limits the field of view of the reflectance, and that's coming from the canopy surface. And so together they can give us this rho value, the reflected radiation. Now, another slide that helps illustrate what happens as we change leaf area index is important. So we can start out with a LAI of 0.25 in this slide. That's the black line. And this slide shows a really nice small difference between a small leaf area index like 0.25 in the red and the near infrared range. And as we increase the leaf area index, you can see how that changes that relationship. But one thing that's really important to notice is that as we increase the leaf area index beyond 3, it's really difficult to tell the difference between the reflectance in the red at let's say 4, 5, or 6, and also the reflectance in the near infrared. And that results in a bit of a challenge when we actually want to measure leaf area index with our NDVI. And that's shown right here. The graph on the right-hand side shows the leaf area index on the y-axis and the normalized difference vegetation index on the x-axis, and shows pretty clearly that early on we can see a large difference in leaf area index, or in our NDVI based on leaf area index. So it's very, very sensitive at low leaf area index. But as we go beyond 3, basically we don't have any ability to differentiate an LAI of 3 versus 5 or 6. It's all lost there in the scatter. But if you look on the left-hand side of the slide, you see a relationship between NDVI and that other index we're interested in, or the other thing we're interested in, which is FI, the fraction of intercepted photosynthetically active radiation. And here we see a considerable difference. And in fact, we're not sensitive to higher fractions of interception. And so we have a nice linear relationship where we can measure the NDVI and actually get the intercepted photosynthetically active radiation, or the fraction of intercepted. So this is a pretty important thing that we can learn from these slides, that in fact we can grab our FI value from our NDVI sensor. Is it as good as our Ceptometer-based measurements? No, I don't think it is. There's a lot more scatter in these data than there would be in a LP80 measurement, for example. But could it be used effectively in the field? Very much so. We can definitely measure this and monitor it over time, and maybe it's a great combination of the LP80 measurements, for example, and a spectral reflectance sensor measurement. One of the things that we can do with this NDVI measurement that maybe we can't even do with an LP80 is leave it in the field to monitor continuously. LP80 wouldn't be a good candidate for just leaving out in the field. Most Ceptometers are too expensive to leave out there, and you'd be concerned that they may get water in them or other things that would limit their use. But a spectral reflectance sensor could be left for the entire season to monitor. And here are some data just to kind of give a primer on what might happen if we took these measurements to the field. This is a study by my colleague Steve Garrity and some colleagues at the University of Idaho, where they were looking at the NDVI as it tracked across a season. These are satellite data that they picked up. And they correlated very well with a lot of things that were happening in this deciduous tree canopy. Start of the season, they're marked SOS. The end of season, the peak of season, the POS, and the beginning of the senescence, the OOS. Those were all points they were able to mark on that curve using this NDVI analysis they did and the modeling of that NDVI across the season. They took those data and then took the derivative of that line and were able to mark several important phenology points along that line, which then importantly led back to an analysis of the resource capture, of the biomass accumulation. They could do from the start of the season, know when bud burst happened, when they were getting leaves out there, at the peak of the season, when they actually could determine when they were basically at the maximum assimilation level. The carbon uptake period was well modeled by the NDVI. So let's move on from talking about fractional interception and the NDVI, the LP80 measurements to this radiation use efficiency value. This is more of a challenging value. You saw I got a lot of data out of the RICE project, but that took a lot of effort and it was extremely complicated to try to get into. So I want to talk about some of the current and recent literature that's come out on work that's shown us how we might be able to use other things to predict this better. But first of all, I've got to say, if you really want to measure radiation use efficiency, you do have choices. Our radiation use efficiency can be, if we rearrange that basic assimilation model that we talked about earlier, our radiation use efficiency is just the biomass accumulation, which we can get by eddy flux sampling, towers, or actual biomass sampling. And then we can divide that by fractional interception, and we can measure it using the LP80 or the spectral reflectance sensor, and multiplied by the solar radiation that we can measure through a pyranometer. And then we get back to that Monteith relationship. We can define the RUE, which will be consistent, we hope, if we're using the same cultivars or in the same ecosystem from year to year, and we can do some estimates then with that information. Now, that may be a little bit rough and just give us some values that are decent approximations, and depending on how we do this, they may not be as desirable as we would like. So what are some other options that we have? Well, let's first talk about some things about the radiation use efficiency where we can just grab from the literature some values and then use those in our modeling. Radiation use efficiency is species specific dependent. C3 versus C4 metabolism, obviously there's a big difference in radiation use efficiency. Legumes have a lower radiation use efficiency, and things with high carbon sink strength, such as tubers, they actually have a bit higher up toward the C4 range of RUE. This is also cultivar dependent, and many of you are aware that crop breeding programs are continually looking for ways where they can increase the radiation use efficiency. Finally, it's environmental dependent, and we'll talk about that in just a moment. The radiation use efficiency is going to decrease as vapor pressure deficit increases. Basically, we got the stomates open, or stomates closed, sorry, try to limit water loss, and also creates challenges for CO2 uptake. Radiation use efficiency will increase under diffuse radiation conditions as well. We saw that, interestingly enough, in our rice crop when during the 1999 season, there were large wildfires in Mexico, which caused some high-level clouds for most of the peak growing season in our South Texas rice field. And we saw that our radiation use efficiency increased considerably. And even though we had more sunshine the year before, interestingly, our total biomass was a little bit higher in 1999 than the previous year. Now, what happens to the radiation use efficiency when things are not ideal? Well, if plants are under stress, radiation use efficiency obviously is going to be reduced. If we're under water stress, nutrient deficiency, if we have problems with disease or temperature stress, these all can change that RUE value. I'm showing again those rice data on the right-hand side. In those conditions, we have very little stress. Rice is one of the optimal crops in terms of if growing with water, it's always flooded, doesn't have that stress. And that flood actually helps with keeping weeds down and other pressures that might cause stress to the rice plants as well. So we can say, I think, that radiation use efficiency is conservative if we're using the same cultivar and that we're not under considerable amounts of stress. Here's a nice little table put together from some scientists at Washington State University here next door by Stockel and Kamenian. They show several ranges of values for radiation use efficiency for different plant types. We have the C3s, obviously lower than the C4 plants. We have the C3 oil crops that are right in the middle, limited range compared to the annual plants. The legumes are obviously quite low. And the tuber and roots actually end up being close to the C4 plants, 1.6 to 1.9 grams per megajoule. Nice table to use. It's something you could use in your research if you weren't able to grab values for the RUE, but you wanted to model the biomass uptake anyway. These are good values to use. Now, here's another study that I quite enjoyed. My father, Galen, gave this to me. It was done by Dr. Bob Kunkel at Washington State University and my father back in the mid-'80s. They were studying the predicted versus actual yield of potatoes in the Northwest. Dr. Kunkel's goal was to try to increase potato production, and he had a lot of plots out there and took, obviously, a large amount of data, and then asked a very important question. He said, well, why is the yield in some of the years not yielding at our maximum predicted yield? Well, that really goes back to the things we just discussed, depending on the nutrients, nutrient deficiency, disease, the temperature, the water stress. All of those come into play and reduce the yield off that one-to-one line there. That's basically the maximum possible yield that we can get out of those potatoes. So we can say that in optimal conditions we're going to reach that line, but anything that's suboptimal, like maybe a limited growing season, for example, depending on the year or something like that, would always reduce that off that line. So some of the studies some of the conclusions that came out of there were that the maximum possible yield varied with growing season length and available radiation, something I just said. Some plots did yield at the maximum. Most plots were suboptimal because of stress and disease, and really came down to limit their ability to capture resources. We also had challenges when there was limited fractional interception, either from a senescent canopy or poor canopy structure if the plant was stressed in any way. For example, these potatoes would tend to reduce leaf area if they were under water stress. So a reduction in leaf area would obviously limit the plant off that maximum line. Higher yields might come from cultivars with higher radiation use efficiency, cultivars with better fractional interception, cultivars with lengthened growing seasons, and the removal of stress conditions to try to optimize that radiation use efficiency. All of these things might help us actually hit that line, or possibly improve over that line if we came up with some kind of cultivar that actually had a better radiation use efficiency. Now I want to get into an area of emerging research. I'm going to throw through a few slides in front of you to kind of help you understand what is available out there. But I want to say that there's several great scientists working on this, but it's an area of emerging understanding, and there's still a lot to learn here. So we're going to talk about radiation use efficiency and the photochemical reflectance index, or the PRI. This PRI was designed to estimate radiation use efficiency by detecting changes in the Xanthophyll cycle. And I'm going to talk about that a little bit more in the next couple of slides, but just what it does is we're looking at how the Xanthophyll cycle essentially is shunting incoming solar radiation, photosynthetically active radiation, off off the chlorophyll cycle. It's taking it into the Xanthophyll cycle, meaning we're getting energy that we can't be used by the chlorophyll, but it's wanting to dissipate that energy. That's what the Xanthophyll cycle does, is dissipate that incoming energy. The PRI, the photochemical reflectance index, is just the reflectance at 531 nanometers minus the reflectance at 570 divided by the sum of those two. Now, why is that important? Well, let's go back a little bit. Dr. John Gammons, back in the early 90s, discovered that the Xanthophyll cycle activity was detectable using a reflectance index, specifically looking at 531 nanometers. So the PRI uses 531 nanometers to measure the Xanthophyll absorbance and compares it to 570, where there isn't any change in absorbance due to this Xanthophyll cycle. And so as that 531 goes up and down, we can see changes in this PRI index. One thing to note is, if we're going to try to measure this, we have to use a fairly narrow wave band, around 10 nanometers for around the 531 and the 570. And on the right-hand side of this graph, we're just showing some relationships between the reflectance at 531 and the epoxidation state on the upper graph, and this also on the xenocyanins on the x-axis, and the change in PRI in the bottom graph, just to help us see that there's a strong correlation between those two. Now, to give us a feel of what's happening, I've put in this graph here that talks about the Xanthophyll interconversion that actually can really be a rapid process. And I've got a couple of slides here to show that as we change the light levels coming into the plant, or the CO2 levels, that the PRI changes, and the net CO2 changes very, very quickly depending on that situation. Now, this graph shows a little bit more in detail what's happening with the PRI. We have the top right-hand graph, and I'm sorry, there's quite a few graphs on this slide, just trying to get a feel for what's going on here. The top right-hand graph shows reflectance on the y-axis and wavelength on the x-axis. And what I want you to notice here is a couple of plants. One is dark adapted, one is light adapted in that upper graph, and it's showing the difference in reflectance of those two plants. From that graph, there's very little to see, really. We look at that, we look at the dotted line and the solid line, and those two reflectances the spectral reflectance looks very, very similar, in fact. But if we look at the bottom line, we look at the delta, the difference in reflection for the reflectance from those two lines. And what we can say is that we can very clearly, once we compare a delta between them, see the Xanthophyll absorbance there coming up at 531, and also the fluorescence a little bit higher. So that's what we're measuring when we're measuring the PRI, is that absorbance there of Xanthophyll. And it's pretty small, and so it takes a pretty good sensor to be able to actually make that measurement. Now, we can also see in a graph here on the left-hand side, I just want to focus you in on the second graph over. We have on the y-axis the normalized light use efficiency and the photochemical reflectance index. I think these for a bunch of Scotch pine that were being measured. And we have a good linear relationship between those two things. Now, here is just some measurements that we made. This is in a grass canopy. And now, instead of looking across a season or taking a whole spectrum, we're just looking at the PRI and comparing it over time, just when we have a cloudy, then a sunny conditions, and then cloudy. So we have highly overcast, then suddenly the sun comes out, and then it's cloudy again. And I want you to see what happens to this grass, that essentially under the overcast conditions, we've got a lot of diffuse light. The grass is happily using that light in its chlorophyll cycle. Everything's going very well, and suddenly we have sunlight hitting that surface, and there's too much light for the grass to use, and we have a large drop in the PRI, which indicates that the fact that we are getting light being shunted in the Xanthophyll cycle, and then when we go back to overcast conditions, the PRI jumps right back up. So you can see a pretty important point here that is, again, I was trying to make this earlier, that this happens very, very quickly. Now, why wouldn't we just want to measure PRI across a whole season and take that as you know, maybe a weekly basis or something like that, and really not focus in? Well, when we look at a hourly basis PRI value, we start to see some of the things that are happening that reduce our radiation use efficiency by the plant. This overcast condition going to sunny condition going to overcast again shows that even though we had a lot more light and would assume that light resulted in biomass uptake, increased biomass uptake by this grass canopy, by the PRI measurements across this day, we really realized that didn't happen. In fact, that increased light wasn't usable by the plants. So let's finally talk about where we can go with all these measurements. Can we make a biomass prediction from our radiation measurements? Well, using our simple assimilation model, we can say that our radiation use efficiency, we know that has some association with the photochemical reflectance index. So we get RUE from the PRI, we can get fractional interception from NDVI, and then we can use a pyranometer to measure the incoming solar radiation. So then can we turn back to that model and get our biomass accumulation? Well, there's considerable data to suggest that's possible. Here's some relationships that were found across a season doing the same thing, but using satellite data, and they produced a fairly good relationship between those two things. Here is using that satellite data, but now mapped across a field. They did carbon assimilation using the absorbed photosynthetically active radiation times the radiation use efficiency, the PRI value, and they were able to map this and show several locations that had high assimilation versus low assimilation and show how that changed across the landscape. Here's a graph done by my colleague Steve Garrity, just showing the leaf area index as it relates to NDVI. Those are well correlated, as you can see here, and that relationship to net ecosystem exchange, which you can see is associated. What we're missing in this particular graph is that intercepted photosynthetically active radiation, the solar radiation, the pyranometer portion, and the radiation use efficiency, although that may be able to drop out of here. So this is a hot topic in research. I don't know that we are to the point where we can say, yeah, this is something you could go out and do. But there's a lot of work that can be done, and I think some good information that can be gained out of any of these processes we're talking about today. From the biomass sampling directly, it's important to ground truth whatever we're doing, to the eddy flux systems or closed chambers that actually can measure the CO2 uptake directly, and now these emerging techniques using spectral reflectance sensors and incoming solar radiation measurements, the pyranometer, to actually estimate our biomass accumulation. So just some take-home points from our discussion today. Biomass capture can be directly measured in a very accurate but somewhat costly and time-consuming way. Using a simple light capture model, we can make estimates of biomass accumulation in the field using our understanding of our measurements that are intercepted solar radiation done by the AccuPar LP80 or by our NDVI spectral reflectance sensor. and by getting our radiation use efficiency, either from tabled values that people have collected over the years, from biomass sampling or eddy flux towers that we've done, or quite possibly by using the latest technology, this spectral reflectance sensor, measuring the photochemical reflectance index. I'd like to thank you for spending a little time with us this morning. I hope this has been useful. I certainly enjoy getting down and studying the latest information that's coming out on estimating and measuring biomass accumulation and in the field, and I hope that you've learned a few things from this seminar today too.